surveillance and early detection or diagnosis of the pathogen is important. And we're looking to also new methods for disease diagnosis, especially AMR, with respect to tuberculosis. And Jonathan is currently a VP of Medical Affairs at Oxford Nanopore Diagnostics and a clinical microbiologist at St. Thomas Hospital in London. His main clinical work over the past 2 decades have been in the prevention, diagnosis, and treatment of ICU infections, particularly those that have been imported and hospital-acquired, and outbreaks in those due to antimicrobial resistant organisms. He's also director of the Center for Clinical Infection and Diagnostics Research at King's College, London, where he leads a research group evaluating translational applications of nanopore sequencing. This spans early research studies through pilot service evaluation of viral amplicon and bacterial isolate sequencing for infection control and respiratory metagenomics for the ICU. His talk today will illustrate how routine nanopore sequencing can reveal important clinically valuable information missed by traditional laboratory methods, which include PCR or culturing methods. It will also look ahead to consider the practicalities of how to embed pathogen sequencing capability into routine microbiology services for patient care, while also providing data for real-time surveillance of AMR and emerging infections. Over to you, John, and we look forward to your presentation.
Thank you, Dr. Sudha, for that excellent introduction. That's it's really wonderful. And, you know, I'm delighted to be here to have this opportunity to speak to you. And thank you as well also to Shivang for organizing this. So I'm assuming you can see my slides now. I'm just going on to the full screen share. And so just to follow on that introduction, to say, to deliver on the ambitions that Dr. Sudha outlined there, you have to work as a translational team between industry, academia, your pathology laboratory, clinicians on the ward to get that benefit through to patients with a real translational mindset. And it was desiring to see pathogen sequencing in service that made me decide to stop doing clinical practice in 2022 so I could fully focus on translation of pathogen genomics. So a few disclosures: we get our funding from a variety of different sources. Our research is done with research ethics, but then when we move into a pilot service setting, we use local hospital governance framework as a new service innovation. And what I want to really, in summary, get across today is, first of all, our experience in our laboratory, which is a newly set up pathogen sequencing facility. We got about £200,000 about 4 to 5 years ago to create a sequencing capability in our routine clinical microbiology laboratory, not a large academic lab, but in a laboratory that was only doing agar culture. And I want to go on and show how once you've set up that facility, that sequencing facility, you can use it to feasibly evaluate a variety of different sequencing tests in a clinical setting. And the key finding is the high frequency of unexpected, unusual, rare, and novel pathogens that you identify when you implement a sequencing capability, which is essentially delivering the promise of pathogen sequencing, and how that really helps bring together people who are concerned about patient care, infection control, and public health in the same time and place, rather than being geographically separated. Now, I think if we take a step back, we all realize that we face an unprecedented and ever-increasing threat from a diverse range of infectious organisms. So there's things that we're in the hospital wards. There's sepsis, that is one of the biggest killer in hospitals. There's pneumonia, which is the 4th biggest cause of death globally, and is the most frequent cause of sepsis. We all know, in our endeavor to treat pneumonia, conditions such as pneumonia and sepsis, we're driving antimicrobial resistance that that is predicted by 2050 to be one of the, perhaps the biggest killer globally. We've just come out of one pandemic. There's a 25% chance, we're told, in the next 10 years of another pandemic. And then advances in bioengineering and AI is increasing the risk of biosecurity threats. The ability to predict using AI the three-dimensional structure of proteins from native DNA sequencers, the availability of benchtop sequencers, makes it feasible that hostile actors and people with bad intent could create artificial pathogens, more virulent and more transmissible, and provide that as an additional threat. And I think actually we need to consider all these together. If we just consider these in a separate box and try and find a solution for each one, it'll take too long, we'll miss synergies, and it will cost too much. We have to ask ourselves, how do we address all these needs in one go? That helps bring funders' attention together. But when you look across microbiology laboratory networks, certainly in the UK, you find that it's not in a particularly good state to respond to those threats. You know, on the one hand, there are hospital laboratories that are often focused on automation through things like Chiestra systems. Hospital labs are being moved off the hospital sites, consolidated into large off-site facilities, still focusing on agar, so it's very difficult to recruit sort of future-facing, talented people into the profession. It's geographically separated from public health laboratories that are looking at surveillance and epidemiology. They're packaging and sending samples across for analysis and sending back results. The hospital labs have also, at least in the UK, they're no longer so embedded in university labs because they're not deemed to be academically high quality. And so there's a separation of research folk from the service labs. So altogether, this operating model is not suitable to address the diverse challenges we face. Now, after the pandemic, the public health laboratory system around the world recognized this deficit and has started investing in pathogen genomics. And so there's reports from the WHO around how we'll address pandemic preparedness, genomic strategies, and antimicrobial resistance. And this includes, for example, large-scale surveillance programs where these public health laboratories gather together bacterial isolates from across the whole region and identify tools for early warning of the emergence of antimicrobial resistance, doing health economic studies to show that sequencing bacterial isolates to understand their virulence and their antimicrobial resistance determinants can be cost effective, and trying to find ways to link that public health sequencing capability into a surveillance program. And actually the nanopore sequencing with the long reads now is ideally suited to very rapidly provide at scale complete genomes, essentially that can identify and separate the plasmids from the bacterial chromosomes. It's not possible with short read assemblies. And also Nanopore have released a full workflow on the community website for nanopore-only sequencing that can help support laboratories in doing this high throughput sequencing. Okay, so I'm going to start with SARS-CoV-2 because I think that's what has really led to the wide distribution and the wider availability of sequencing capability in clinical and public health labs around the world. But of course, as SARS-CoV-2 went away, a lot of those sequencing capabilities were decommissioned. And that includes in our public health labs, and the reporting was all decommissioned. But we opened a epidemiology SARS-CoV-2 sequencing capability in our clinical laboratory. And so we kept going with that sequencing after the pandemic had ended, and actually thought that that was an important and useful thing to do. So in 2021, we wrote to the Lancet Infectious Diseases because when we asked at the beginning to be a clinical lab contributing to public health surveillance, we were the only one in the country, and they were a little bit uncertain about whether a clinical lab would be up to meeting the volumes and the needs of a national program. And so we just highlighted when we identified a vaccine escape mutant in 2021, on a sort of same day or a sort of 24-hour turnaround hospital embedded SARS-CoV-2 sequencing service, that there was utility in having that capability on the front line. And then in 2022, when actually the main cases that we saw with SARS-CoV-2 were in our immunosuppressed patient populations, we published this report that actually had high impact in clinical infectious diseases, where we were demonstrating how not only is SARS-CoV-2 sequencing helpful for the epidemiological sort of global public health perspective or for hospital outbreaks, but could also be helpful in individual patient management. And I've got 3 examples here where of immunosuppressed patients. So this is a 50-year-old woman with thymoma who had LFD positive COVID in February and April 2022, and then arrived in June 2022 to have a thymoma removed, but was still SARS-CoV-2 positive. And so we sequenced that isolate, and it was the Omicron BA.1.1 isolate that was no longer prevalent in London. So that was indicative of chronic infection, appropriately treated with agents that were no longer being used, PCR negative, and that allowed the surgery to proceed, thymoma removed and discharged. Whereas if we hadn't done the sequencing, we would not have been able to predict which therapy to use to make the PCR negative. It's an example here of a 54-year-old woman with HIV who had chronic asymptomatic SARS-CoV-2, was treated with Regeneron in February, but 19 days later was still positive and was becoming symptomatic. So we sequenced and found they had mutations conferring Regeneron resistance, so was treated with Paxlovid and went PCR negative. And then there's lineage calling when there's lots of different lineages around. 47-year-old woman waiting liver transplantation, was COVID positive, couldn't go for the liver transplant. BA.5 was dominant in London. We sequenced the patient. She was BA.2.12.1, which requires a higher dose of sotrovimab, so was given double dose sotrovimab. So that's an example there I'm showing where we use SARS-CoV-2 for global epidemiology and local epidemiology for outbreaks, and also for infection, for managing the patient. And then in 2023, we were one of the few labs that was still doing routine SARS-CoV-2 sequencing, and we weren't reporting them to UKHSA anymore. So Luke, my PhD student actually, went onto Twitter, now X, and said, look, I've just found the 5th case of a new highly mutated BA.2.86 in our hospital, and we're going to send it through for culture. And he did that early in the morning, and at 8 o'clock, the National Incident Director was phoned by media saying, so what are you going to do about the emergence of BA.2.86 in the UK? And the Incident Director, who I know well, said, well, we don't have it in the UK. They said, yes, you do. Somebody called Luke Snell's just identified it. So actually, they're a bit annoyed at us for having done that, the clinical lab reporting through Twitter. But actually, that was helpful because it brought forward the UK's vaccine program due to their identification in the UK. So this is another example why having your sequencing capability close to the front line in clinical laboratories not only helps patients, but can have profound implications even through to vaccine strategy. So let's now move on to example number 2, which is flu. So Tom Williams here was a registrar that worked with me when I was providing infectious disease consults on the ICU during the COVID pandemic. And he heard about some of the work we were doing, so took a year off to come and work for us. And he set up a 3 times a week, real-time Nanopore Influenza A sequencing service in the clinical lab, predominantly during winter, and that's the epidemic curve during winter, to provide support for infection control. So we sequenced all the flu isolates in the swabs that came into the hospital over the winter, so that we could build up a sort of a phylogenetic tree. And then when the infection control saw cases of hospital-acquired flu, we would sequence those, and we identified and communicated or excluded the presence of clusters of identical flu strains on those wards that could help target infection control, closing the ward. And because we had the influenza A sequencing service up, we could also then apply that to other settings. So this is an example of a patient who'd had a failed renal transplant and was on hemodialysis and was flu positive with wild type flu A. But They were not clearing the influenza, despite having oseltamivir after each dialysis session. And the CT value went down on PCR. So we sequenced their isolates and found that they had acquired some a mutation in their neuraminidase that codes for oseltamivir resistance. And so that was in requiring a zanamivir treatment. And then also we had a gentleman admitted from Southeast Asia with flu A that was untypable. And so that person was removed to our high consequence infectious unit because according to UK guidelines, somebody admitted from Southeast Asia, we need to exclude avian influenza. This was in an untypable flu. And we sequenced that within 24 hours. And it was actually H3N2. They could be treated with oseltamivir and released from a side room. So again, similar theme. You introduce a sequencing program in your clinical lab for an high consequence organism, an important organism, and you can see how you have benefits for infection control, treatment for the patient, and for public health purposes. And also you get surveillance of the flu variants that can be helpful for informing national vaccine programs. The third thing I'm going to talk about now is the isolate sequencing. So again, because we had the Nanopore Sequencing lab in situ, and we had 2 laboratory assistants, a clinical scientist, and a PhD student, we decided to see, we wanted to see what the clinical impact could be of opening offering a prospective isolate sequencing service for infection control. We did that last winter, over 4 months. We had infection control team contact us about 13 suspected outbreaks on the wards, and we sequenced 61 isolates from 42 patients and 5 environmental isolates. We would return those results within 48 hours of request, another advantage of having that sequencing on site, rather than needing to send them off to a typing reference lab that could take 10 days or 2 weeks. A third of suspected outbreaks could be refuted. The cost per isolate in terms of consumables was relatively reasonable. And essentially what would happen is you would gather the agar plates after you had a phone call from infection control. And after a 2-hour extraction protocol, load on the flow cell, sequence overnight, and then give them the report the next day. Now, you'll probably note, those of you who looked at the sort of public health sequence isolate sequencing programs, that they're really focused on antimicrobial resistance. And so they target their focus on sequencing resistant strains. But actually, that's not always the case. This is an example here where I was on a ward round and I saw a 40-year-old man who had a discectomy in their back. And they were mobilizing, walking down the stairs. They had pain in their back, and the wound opened up and pus came out. And it was cultured, and it grew Pseudomonas aeruginosa. So it was fully sensitive. We started them on some treatment, but I thought that's a bit odd. We went down 2 floors to another ward, now to the orthopedic ward. And there was another patient there, 76-year-old woman, who had a left total hip replacement. And I said, well, look, it's often infection control and microbiology is a bit of a detective stories. And I thought, hang on, 2 patients, Pseudomonas in wounds. Admittedly, one was in the thoracic service and one was in the orthopedic service. That's a bit odd. So went to have a look, see where they had their operation. They both had their operation in theatre 4. So try to receive both isolates. This one was saved because it was in pus. This one had already been thrown away by the lab. But we got some theatre water samples, and we sequenced isolates from the theatre water and from the patient, and they were both ST309 with only 10 SNPs difference across 5.8 megabyte genome. So a serious incident was declared. Water filters were fitted on the taps. We did a retrospective analysis of all Pseudomonas in wound isolates on that hospital. That generated quite a lot of interest, actually. And when we had 3 patients with sterile Cytochromobacter infections on ICU over 4 months, the intensivists were saying, well, perhaps we've got a waterborne Achromobacter outbreak. So we sequenced those isolates and they were all different. So we could exclude a point source there. So that it illustrates, that sequencing is helpful both to exclude and refute cases. Okay, so those are examples of targeted sequencing programs, isolate sequencing, Amplicon. And they show, I think, all of them now. I think this is a common theme, and I think it's what we'll always find. When you introduce sequencing, you bring together the utility for the patient care, for infection control, and a wider public health. I'm now going to turn to metagenomics, and the last part of my talk now is going to be focused on that. Because I think this is probably the most exciting project that we have been involved with, the one that we're taking the furthest. It's the one that holds that promise of that agnostic, open-ended question of sequencing everything in a sample. And just to say what it is, what I've been showing before is where you just target one bug. What this is doing basically is saying you sequence all of the organisms in a sample, and that includes, that then provides you with identification of the organism, the presence of antimicrobial resistance determinants or virulence determinants. And if you have sufficient numbers of organisms and you sequence for long enough, you can get the whole genome as well for typing. So I'm going to talk about the work that we've done over 4 years, from a laboratory workflow development phase published here, clinical research, pilot service evaluation, and then this paper I wrote a year or so ago, trying to identify a roadmap through to implementation and adoption. So I work at St. Thomas's Hospital. This is how our ICU network is for the adults is arranged. We've got a medical ICU on one floor, surgical on another. Then we have an ECMO unit, extracorporeal membrane oxygenation, where the blood from patients with severe respiratory failure is oxygenated in a machine, a bit like a dialysis machine, before being pumped back in. That's in a high consequence infectious diseases unit. And I put those blobs as red, green, and yellow just because, you know, this is what happens. You go around the ICU, some people are well and almost ready to go home. So they're elective surgeries. Some people are really unwell, some in side rooms, some are perhaps recovering from infections or starting to develop something. And each day that color map changes. The patients are different each day. Some have left, some have died, some are better. And that's why, you know, you need the results that day. The day you go down and see that map, that's when you need to give them. You can't go along and say, well, I can't tell you what's in the samples on the patients you're interested in today, but I can tell you the ones that you sent 3 days ago. That's what we currently do, and that's just not acceptable. That's on the right there is the various tests that we do. You get quite a lot of tests when you first come in, but after you come in ICU acquired, essentially it's largely just respiratory. And the ICU does a respiratory 16S test. I'm not quite sure why. It takes 5 to 7 days to come back, and they look at them retrospectively. But that was our inroad into developing a nanopore sequencing test, because we said, you're currently doing 16S on your respiratory samples from these severe respiratory failure patients. We're going to develop a same-day, on-site nanopore sequencing 16S test. We spent 3 years doing that. We divided the work into 3 phases. The first is the research phase, where you develop the assay and show that it works against all the different isolates that it needs to. The second phase is where you validate that against the routine tests done in the laboratory, using research ethics on stored respiratory samples, developing your training program, a validation data set, a standard operating procedure, and get yourself set up to then go into phase 3, which is where we use we get permission from the critical care and hospital governance program through the quality improvement and patient safety scheme around the logistics and the resulting and of providing a pilot service where the intensivists would send the samples down in the morning, and then in the afternoon when I was on the ward round, we would have that 16S result from the respiratory sample, and then carefully document all the impacts and look for safety signals. And this has already been published. Rosella Bolden was the PhD student, postdoc, who led this work. And actually, I won't go into the details. In about 2/3 of cases, antibiotics were changed in response to the results. But we were given to providing the service in March 2020 when we had to stop because of the COVID pandemic. And we stopped partly in terms of the COVID pandemic, but also, you know, there's the recognition, although this worked well, it doesn't provide antimicrobial resistance genes, and it doesn't provide, it doesn't detect fungi. And also in March 2020, Rosella Bolden left and a new postdoc arrived, Themis Charalampos, who'd been working with Justine O'Grady in the University of East Anglia. And she had just published this paper. She'd spent 3 years in her PhD doing the research phase around about a 7 to 8 hour end-to-end workflow where you take bronchoalveolar lavages, you deplete the host cells using a saponin-based lysis of human cells, DNase treat to lyse the released human DNA, and then the remaining intact organisms, bacteria, apart from those without a cell wall, most fungi, but it doesn't seem to work for PJP, and some DNA viruses are then extracted, and there's lots of different extraction machines. We use a Promega, a Roche MagnaPure. And then you use a low input rapid barcoding kit PCR amplification phase to create a library that you then sequence. So we sequence either up to 8 or 10 samples on a MinION flow cell on a gridiron, or single samples on a flongle. So that was the basic science that Themis had done. So during that first wave of the COVID pandemic, when our ICUs expanded from about 41 patients to 250 patients across the pediatric ICU and the respiratory wards and the cardiology wards, I suspect an experience that's familiar with people all over the world, actually. In the afternoons when I was on those wards going around, I identified new people with suspected new secondary infection on top of COVID, alerted the clinical lab, who anonymized the samples, and then they were handed over to the research lab, and they were batched sequenced on flow cells with an 8-hour turnaround time. Good performance characteristics, detected beta-lactam resistance genes, beta-lactamases, and we had 100% concordance for Aspergillus fumigatus. And these were in an 8-hour report, 8-hour end-to-end workflow. But actually, we continue the sequencing overnight for 16 hours. And that allows you, as I said before, to construct the full genomes of the metagenomic output. And this is really a table showing the Klebsiella pneumoniae. It was an ST307, a multi-drug resistant Klebsiella. It wasn't carbapenemase resistant, but it had quinolones and other beta-lactamases. And most Klebsiellas in this sort of situation will differ by thousands of SNPs. They're clearly unrelated, but there was 2 samples here, and in fact 4 in the end, that only differed by a few SNPs, highly suggesting relatedness. And then we went to one of those wards during that May time. We found those 4 patients overlapped, so the sequencing overlapped with the epidemiology. So that's that proof of principle that you can use a respiratory metagenomic test, but both with satisfactory performance characteristics, but also for the epidemiology. So we then went through the process of, exactly as we did with the 16S, of taking that metagenomic assay through into a service setting. So we generated an automated bioinformatic tool that basically, as the sequence comes off the gridiron, you remove the remaining host DNA, and then within 30-minute sequencing, you can provide an identity report on the organisms with an estimation of abundance, and that's then signed off by the clinical scientist to produce a clinical report, to confidently identify or exclude AMR genes using SCAG-AIR. You need 2-hour sequencing. We needed 2-hour sequencing to generate laboratory reports. There'd be a follow-up report. And then we had a manual approach to the genome assembly, MLST, and SNP analysis at 16 hours, reviewed by the clinical scientists. So that was the bioinformatics. In terms of the reporting, this is what a metagenomic output looks like. This is an example of a young chap who had a car injury with multiple fractures and lung injury. And this was on admission, Prevotella fusobacterium, so Prevotella, lots of anaerobes and Staph aureus. We now, in going into a pilot service, you spike each sample with an internal positive control, a bacteria that you'll never see in clinical samples, Jonessia, and we spike that into a limit of detection, and we set reporting thresholds based on a validation set. And that's the report that you give to the clinicians. And at culture, it just identifies the scanty Staph aureus, no anaerobes. So then we proceeded with the satisfactory validation reports with our QC criteria. We went into a same-day pilot service under our hospital governance program, winter 21-22. Actually, that was a time when they were predicting a third wave COVID pandemic, and also resurgence of flu. And so we wanted this service to help us manage the secondary infections on the ICU. Now, as it happened, we didn't see a significant COVID-19 resurgence, and there was no flu that winter. But we proceeded ahead for a feasibility study, clinical impacts, under a defined SOP after a 3-week pre-pilot shadow service where we did the sequencing daily but didn't report back. And then we would record the prescribing decisions of the treatment decisions made by the intensivists that they made themselves. They chose the patients in the afternoon. And then we reviewed all cases at a bi-weekly review to detect any safety signals, of which I would say we didn't have any during the pilot. So this is the headline results. We've done over 200 tests now. We're getting an average sample to the first result of 6.7 hours, 90% getting same day. We change antibiotics in about half the cases, and 20% escalate because they're on the wrong antibiotic. 25% cases we de-escalate because they're on broader spectrum than they need to be. Also suspect there's benefits without an antimicrobial change. Often clinicians want to escalate antibiotics or antimicrobials of some description because they don't think the patient's getting better fast enough. We could do the metagenomics that would reassure them that either there was no organisms or no new organisms, so that prevented escalation. Then also in this complex patient set population, sometimes the view is that it's due to an aberrant inflammatory response that's causing the infiltrates in the chest, and they want to immunomodulate. But normally they wait 2 to 3 days. We're waiting culture results because you know just to make sure there's no infection there. But this would bring forward the immunomodulation, and we think is a really key benefit of a rapid same-day metagenomics service. Then also there was, a spectrum of unexpected and unusual things. And I'm just now going to spend the last 10 minutes talking about some cases just to illustrate. So I'll go through these quite quickly. I'll just highlight on the key points. This is time. So patient comes in with flu, 3-day short history, ends up on ECMO, very inflammatory, BAL taken on admission, 5 o'clock in the afternoon. This is the report. It's got Staph aureus in it, mostly some strep pyogenes. Also the 30-minute report, it's got AMC, that's macrolide resistance, MEK-A, it's MRSA, fusidic acid resistance, and LUC-FS, it's PVL. So the report to the clinicians the day of arrival tells them what the bugs are. Actually, they hadn't started IVIG. They did in response to this information. And the infection control public health team were informed this was an MLST ST152 the following day. Culture arrives with the result 5 days later, telling us it's MRSA. 2 weeks later from the reference lab that it was PVL. How about that strep pyogenes though? Well, because we had that isolate sequencing service running, and this is the thing, once you have your nanopore sequencing capability set up, whenever there's a new question or a new issue, you can apply it to a different situation. So we had UKHSA report a national epidemic of group A strep infections, winter 22-23. I mean, that was all the other previous 5 years. This was last winter. It was a very bad, actually, with some pediatric deaths. So as soon as we heard of that on the 6th of December, we retrieved all of our group A strep isolates over 2 weeks to look at the clinical risk factors and the genomic risk factors for severe disease. And in a multivariate analysis, identified it wasn't the particular clonal type that was thought to be the association with severe disease. It was actually carriage of these two superantigens that's been confirmed in other reports now. So we were very quickly able to support the national lab with that identification of group A strep. Now that was December the 6th. We're then saying, well, remember we had those cases, because there was 3 or 4 of them, back in October, where we had some group A streps and they were with other organisms and we didn't look at the sequence. We then went back and constructed the full genome of those strep pyogenes. And I found out it was exactly the same clone in October in these ECMO patients that was causing the disease later in the year, giving that indication of how, with a metagenomic sequence, first of all, the data is saved. Imagine going back and saying, could you get out that group A strep that you isolated 3 months ago? Of course we haven't. It's been thrown away. Whereas, of course, the data is collected forever when you do sequencing, and therefore that can help you to early indication of new infections. And we had another patient with flu come in actually 5 days after that previous case in October. They were also really very unwell with a high CRP and a PCT, did a BAL, and we reported Streptococcus dysgalactiae, group C strep, and a Staph aureus PVL again. And just like the previous case, the culture only identified quite late the Staph aureus and the PVL 2 weeks later. But at the time, we didn't look at the Streptococcus dysgalactiae any further. But after the group A strep had been identified, we said, well, let's have a look at that organism too. And it turns out to be a Dysgalactiae subspecies equisimilus, which is an emerging highly virulent group C strep reported in Scandinavia. Another example of how the pathogen sequencing identifies things that your culture doesn't identify and gives you the full picture of its concern. This was a patient with flu, a 30-year-old woman who failed conventional ventilation, very not inflammatory at all. Nobody thought about a secondary infection was a concern. It was just on tazocin. We did the BA 0640. Metagenomic report in the afternoon was all above threshold Aspergillus fumigatus, so much so actually that we could construct the full genome, and they could then start on amphotericin and did well and came off ECMO. And it's not just the community-acquired infections. This is a patient here who'd had an operation, was extubated and then got re-intubated and developed a temperature and a pneumonia, but because she was penicillin allergic, had gentamicin and vancomycin. We did the respiratory metagenomics on that sample, and lo and behold, and to the surprise of all of us, we identified Legionella pneumophila. Again, from the metagenomic output, we could identify the type that was also confirmed by the reference lab, and we took water samples from the sinks around that side of the ICU and found high titers of Legionella pneumophila of the same ST1326 type that was in the patient. So a serious incident was declared, filters were fitted, and presumably other cases were prevented, and the patient did well and left. This is a 68-year-old man with a complex thoracoabdominal aorta replacement, massive transfusion, no history of immunosuppression. He was extubated early, but then reintubated, was escalated through up the antibiotics. Culture only showed Candida albicans, so was started on an nodule fungin. But around the third week, day 21, it's really not doing well when the light and sedation was clearly obtunded, liver function tests going off, platelets were low. And we did metagenomics on the respiratory sample, and it came back as pure only herpes simplex virus 2. And so we started them on high dose, the patient on high dose acyclovir, concerned that that was significant. Also had a small rash on the abdomen. The intensivist asked the dermatologist to attend, who thought that the BAL-HSV was not insignificant. They probably had a cold store and suggested stopping the meropenem. We thought, however, that it was, and so we took a plasma sample and a vesicle swab and the wound swab, and they all had HSV2 in them. So this was a case of disseminated HSV2 revealed by the metagenomics. Okay, so that's the end of the case series. I would say, you know, one of the shortcomings of that particular workflow, as I mentioned before, was it doesn't detect atypical bacteria or RNA viruses. So we've got a mechanical host depletion test that we're setting up now that takes about 6 hours, that has been running in service this winter. Over 100 samples have been processed, and that's going well. And I'm just illustrating here how in a sort of unselected adult and pediatric severe pneumonia cases, direct on the respiratory samples, these are the CT values by PCR. You can construct often full genomes to get the types of the SARS-CoV-2. You get a range of respiratory viruses, even respiratory viruses that you don't often see, or certainly not in our panel, influenza C in one patient. And overall, in summary, assemble the genomes in 42% of viruses and 30% of bacteria. So when we had the international Mpox outbreak, actually, with patients seen in our high constant infectious diseases facility, cases were increasing in the UK. There wasn't a local PCR. So we got health and safety agreement to swab the patients on the ward at that time and got a full first full UK genomes within 7 hours of processing the samples. 2 had Mpox and 1 actually had varicella, so it could be removed from isolation. I think I'm going to stop there because I'm conscious of time. And just to say, given the success of the respiratory metagenomic program, we've had funding from the NHS and government to roll out this metagenomic sequencing capability at a number of sites around the UK, starting in December last year. So we're supporting Newcastle and Birmingham, Manchester, Oxford, Southampton, Great Ormond Street and Cambridge to get set up with an equivalent metagenomic sequencing capability, with the data from each of those sites being sent up to our public health department so that they can get this real-time perspective of emergence of concerning organisms or AMR resistance. And so that's, I think I'll stop there. And so I've shown some evidence here that it's feasible to demonstrate a wide variety of clinical impacts by introducing a pilot nanopore sequencing service. I think there's benefit in setting it up. I think different labs in different cities, different countries, you'll find different things, but I'm sure there's things of value that you'll find, particularly highlighting this hidden burden of infection missed by our the current techniques that we use. I think you do need some investment in the sequencing laboratory to start, to set off, and I think you need collaboration between your clinical teams. It's good, helpful to have some academic trainees who are doing some research programs, healthcare scientists. You do need to be prepared to overcome hurdles and barriers because it's very disruptive. The notion of introducing pathogen genomics, and bring your skeptical colleagues with you along on the journey. But looking ahead, we certainly need clinical trials now to demonstrate clinical utility and health economic benefits. We need to automate and standardize these techniques and think through laboratory accreditation and take these tests through accreditation. So I thank you very much, everybody, for listening.